with this super resolution microscopy. So I want to now go on to the next signpost. This super resolution imaging is really an amazing new window into nanoscale structures in cells, 5 times, 10 times beyond the diffraction limit, and various people are coming up with even more ideas to push the resolution even further. But what does this do for us? It allows us to have a deeper understanding of cell biology, but also catalysis. People are using some of this for catalysis studies. Materials, polymers, shapes, and polymer mixtures or block copolymers. All of these interesting systems that have detailed structures below the diffraction limit can be observed using super resolution if you can label what you want to see. And these methods may lead to some important applications. For example, it's not only worth studying the normal cell biology, you can also study pathogenic cell biology. What happens with amyloids, what happens in cancer, and so on at this very, very tiny scale. So now, I want to talk a little bit more about one special aspect of these experiments, which is 3 dimensions. We'd like to be able to do all of these imaging tricks, including the Z dimension, not just X and Y, because you know the answer. The world is 3-dimensional, right? It's not 2-dimensional. If I'm trying to study some property like the motion of this transmembrane protein on the surface of a bacterium, but I can only observe it in 2 dimensions, you see that I only observe part of the motion, and miss a lot of the motion. So we've been working on techniques for 3-dimensional imaging, and here's one of them. So this thing on the left actually gives you, it's going to be a mind bender a little bit for some people. On the left is a standard microscope where all I'm doing is I have a single emitter and I'm moving it to different Z positions, and you see it's in focus at one time, but then it goes out of focus and it disappears very quickly. That doesn't help you very much. We change the microscope by just putting some simple optical components into the microscope. A little glass disc that has some different thicknesses in it. And by some beautiful magic, of optics and all of these things that you've heard about, it actually comes from differential equations and the waves of light and so on. But anyway, a single emitter becomes 2 spots. 2 spots. And now if I move it up and down in Z, those spots revolve around one another. And that angle between those 2 spots gives me Z information. So this trick takes a conventional microscope and gives you X, Y, and Z. So you can use that to get 3-dimensional images like these, where the surface of bacteria are colored one color, and there's a protein inside that's colored with a different color. And this is really just playing with the light from the molecule. Since we understand how that light works, understand how it behaves as it propagates through space and so on, you can make these sorts of changes, and you can even make a single emitter look like a happy face, believe it or not. So here's one thing my students did. You've got some single emitters, and now we turn on this special optical component, and you see that all of a sudden no, please don't tell me. Why is my video not playing? Never allow a place to demand that you not use your computer. So these single molecules turn into a happy face, and then as you move above and below the focal plane, they turn into an unhappy face. Like my face right now. So using these 3-dimensional techniques, you can now look at structures inside bacteria on the left and see the full 3-dimensional shape of a helix-like structure. You can see structures at the ends of cells that certain proteins make. And on the far right, I'm hoping this one works. Does it work? The nuclear lamina? Yes, that one works. This is the nuclear lamina in a mammalian cell. So in almost all of our cells, there's of course a nucleus and a nuclear membrane. Right inside the nuclear membrane is the lamina. This is a 3-dimensional super resolution image of the lamina, where you can even see a channel from one side to the other of the lamina itself at super resolution. So now, the last thing that I want to point out and just remind everyone so no one's confused, I've been talking partly about imaging of single molecules to produce a three-dimensional super-resolution image. That's an extended structure, and it needs to not be changing while we're measuring it. We want to measure that extended structure, and I've shown you many examples on the left. In turning that diffraction limited into this rotating image, which is not rotating. There it is. Okay, but let me just emphasize that this trick of three-dimensional information also works for single molecules, of course, at low concentrations. So here I'm showing you what happens on the right with this thing that we call the double helix point spread function. And I'm showing you the motion of an object in 3 dimensions, even though the movie's not playing. That is a single RNA particle, actually, an RNA particle inside a cell nucleus that we can observe moving over a long time scale. And that kind of motion, of course, tells you about what's going on in the cell. Let me emphasize that a little bit more. If I can watch the single molecule move, then I will learn the science by just simply following its motion, measuring its motion, and understanding its motion. So here's one of many examples of this idea. A little bit more biology. In all of our cells, on the left, we have a structure called the primary cilium. The primary cilium sticks out from the cell, and it's a signaling center. It contains proteins that are involved in something like differentiation and other signaling between cells. So I'm showing you how a protein can come and bind and change the behavior of the cilium and actually change the genetic program of the cell. So this is a crucial process in differentiation, and also a crucial process in cancer, apparently, because these particular proteins become mutated quite a lot. So on the right, I'm showing you that we can image a single molecule. In this case, we're talking about this one that's green on the left. It's called patched, and a single patched in the cilium can be observed moving around and superlocalizing its position. And at the bottom, I'm showing you its position as a function of time, where the motion is part of the time gray, which is diffusion, part of the time is directed motion in blue, part of the time is binding in red, because it's moving and finding partners and doing things. It's being pushed by motors sometimes, other times it's moving by diffusion. All of that is hidden in the motion of one molecule. And we have a great challenge now to get as much information as possible from these kind of data, which involve mathematical analysis, statistical analysis, using methods that can extract that motion dependence in as much detail as possible. So I'm very excited to have had a chance to explain to you this field of single molecule spectroscopy and imaging and super resolution. The field has had impact in chemistry, in all the ways I list here, and others, in physics and in biology. And working in this field has been extremely exciting for me over the past 30 years, because there are scientists everywhere in this global scientific enterprise that we're involved in who have made important advances to the field. I want to thank my past students, postdocs, and collaborators. And here's the current team. Here's our money and so on. At the lower left, you see a little logo, no ensemble averaging. If you like that. And I say that we're the guacamole team, right? The guacamole team. Well, what's a guacamole? It's one molecule, obviously. One molecule. 1 over avocado's number of moles. So with that very, very serious and sad joke, I want to thank you again for your attention and celebration of the Mendeleev 150th year's anniversary.